What's up guys, Omio here. As you have already heard about the coronavirus or COVID-19, you know, the quarantine, the disease, etc. It's like everywhere in the news, everywhere you look, it's like coronavirus. But you probably don't know much about its biological aspects, like what it looks like, how it attacks your body and replicates and etc. So we will take care of that in this video. Coronaviruses are a large family of enveloped RNA viruses. A subset or subclass of these viruses now cause diseases in humans. Now 7 of these coronaviruses are actually known, SARS CoV-2 being the 7th. So here, SARS CoV-2, as I said, being the 7th virus in this subset, causes actually the disease COVID-19, which stands for Coronavirus Disease 2019. It is also sometimes called the novel coronavirus because this virus is actually brand new. We haven't seen this one before. Under the microscope, the SARS CoV-2 looks like this. The new coronavirus, the novel coronavirus, looks like this under the microscope. So this picture has been published by the Robert Koch Institute in February, which are a leading research institute worldwide. They are actually doing a pretty good job with it. They're trying to study it more closely in order to be able to make a vaccine against it as quick as possible. That's why I'm also trying to get in there for my internship because they're really good. So wish me luck. Corona is actually derived from a Greek word which means crown, because these viruses under the microscope look like a crown. As you can see in this picture. Oh, not this crown. Not this one. Yeah, this one looks good. Oh, okay, sorry. So they look like this because of their spikes, like a crown. That's why they're named Corona. Now we're going to be talking about the anatomy of the new virus called SARS CoV-2. We're going to talk about its shape, its size, its molecular biological structure, and the components of its structure. The SARS CoV-2 looks very similar to the other coronaviruses we know in this subfamily. There is no prominent structural difference between them. If you look at the MERS, SARS-CoV-1 or the SARS CoV-2, the latest one, everyone looks the same under the microscope. But there surely are some biochemical differences in some components of the SARS CoV-2 which differentiates this one from the other coronaviruses in this subfamily, which we are gonna talk about later in this video. So the SARS CoV-2 or any other virus in this subcategory have a roughly spherical shape with a size ranging from 80 to 120 nanometers. But sometimes the viruses can also be as big as 150 nanometers. Just so you know, because of this size, because they range from 80 to 120 nanometers, you can prevent them entering your body through a mask, because a mask normally is porous, and the pores of a mask range from 0.1 to 10 nanometers depending on what type of mask you have. So that means any type of mask you're wearing, it can prevent these viruses entering your body through your nose or your mouth. You can see the structural components of this virus or any other virus in this subcategory. You can see a nucleocapsid, membrane, RNA, envelope, and spikes. Those are actually the main components of a virus. Let's take a look at the functions of these components. Let's start from the RNA, because everybody knows what it does. It contains the genetic information for reproduction once it attacks its host. The E protein forms the envelope, which protects the viral genetic information. So basically, it's a protective layer. Spike or S proteins make the spikes, which are glycosylated transmembrane proteins up to many thousand amino acids. These amino acids have 2 to 3 domains, mostly N and a C-terminal domain, which are also called S1 and S2. And these domains are actually responsible for the binding and fusion of the virus to the host cell receptors. The N protein binds the viral RNA in a beads on a string fashion to the capsid, thus forming nucleocapsid. So basically the nucleocapsid is just a protective coating of the RNA. The N protein here also has many domains with different rests. And because of these amino acid rests in the protein domains, it is able to attach itself to the RNA. The M glycoproteins or membrane proteins are the most abundant components of a viral membrane. It forms the shape of the envelope and provides structural support to it. It also plays a role in providing immunity to the virus. Let's talk about the genome of this virus. It is a positive sensed RNA virus, which means that the RNA strand runs in 5 to 3 direction, kind of like same as the messenger RNA in a normal cell. And by a normal cell, I mean a cell that has been, or a DNA strand that has been transcripted. And its genome consists about 29,861 base pairs. How does the SARS CoV-2 replicate itself when it's inside the host? SARS CoV-2, much like any other virus, are highly specific, which means they attack the cells that have that particular receptor that they can bind to. Cells that don't have that receptor face no attack because the viruses can't bind to those cells. The specific receptor that the SARS-CoV-2 can bind to is the angiotensin converting enzyme 2. So the virus attaches itself through its spikes on the receptor, and so does the replication of this virus begins. After the attachment of the virus to the ACE2 receptor, the virus is engulfed into the cell, just like endocytosis. And remember, biologically the replication has started at this point, so in those terms the person is now infected. After the engulfment, the deposition of nucleocapsid occurs through a series of biochemical reactions. The viral genome, the RNA, is now ready to be translated. One of the first segments to be translated is a complex polyprotein replicase, also known as RDRP. This replicase complex then uses the genomic RNA as a template for more RNA synthesis, means it replicates it, and so a new copy of RNA is made. But the translation doesn't stop there. The production of E, S and M protein synthesis also takes place. These structural and other accessory proteins are then sent to endoplasmic reticulum, from where they transit to ERGIC, which stands for endoplasmic reticulum and Golgi intermediate compartment. Proteins at this point are N-functional proteins, which stands for nucleocapsidal proteins. So they are packed polypeptides at this point, which after exiting the ERGIC encapsulate the progeny genomic RNAs and form nucleocapsids. These newly formed nucleocapsids then coalesce with the membrane-bound components, forming a new virus. But remember, not all the structural components are formed during the translation. Many of the host proteins are assembled to make structural proteins like membrane or spike proteins for the newly formed viruses. At this point, the progeny viruses are ready to attack other cells. The newly formed viruses leave the cells mostly via exocytosis. But in some cases, the host cells also die because they get damaged really bad, and all the newly formed viruses get released into the environment. Once the viruses are released, they are ready to attack the new healthy cells. You can see the whole replication process in this diagram from the start, how the viruses attach on the receptor, and till the end how they exit the cell. But wait, sometimes you're just having a bad luck. What happens is, sometimes some fractions of the processed S proteins that haven't been assembled and are still present in the host cell, they assemble and cause a fusion of cell membranes between adjacent cells. In this scenario, infection of a huge cell occurs, which leads to a huge release of progeny viruses, causing more damage to the tissue and the organ. When the virus has invaded thousands of your cells and the infection is spreading, it moves to your other respiratory tract organs like your mouth, like your throat or lungs. Your lungs get more damaged as compared to your throat or other respiratory tract organs because they have more ACE2 receptors. You might think now, okay, but there's not just one type of cell in lungs, there are many types of different cells. But yes, all of them have more ACE2 receptors as compared to your other organs. That's why your lungs get attacked that much and they're so sensitive to this virus. One type of cells in particular is very sensitive to this virus. These are the alveolar epithelial cells. When these cells in your lungs get infected, it leads to lung inflammation, which makes it harder to breathe, which then leads to pneumonia and infection of alveoli, the air sacs in your lungs. At this stage, the mucosa collects in your lungs, which then blocks the carriage of oxygen to your blood. Less oxygen in your blood, that means less transport of oxygen to other organs, which can cause tissue damage in those organs and eventually lead to an organ failure. The key to tackle this epidemic is to make a vaccine. I mean, that's not a secret, everybody knows that we need a vaccine. But why are we not able to make a vaccine? To be able to make a vaccine against the SARS-CoV-2, we first need to understand its receptor recognition mechanism, which regulates its infectivity, pathogenesis and host range. A lot of research centers are focused on this point at the moment. They are trying to study and understand more about it in order to fight it. The more they know about it, the quicker they can develop a vaccine against it. The SARS coronavirus and the SARS coronavirus 2 both recognize the same receptor, the ACE2, but the binding ridge in SARS-CoV-2, in the novel, the new one, takes a more complex conformation, and also it has some additional stabilizing structural residues which were not seen in SARS coronavirus 1. So the understanding of the more complex structure of the receptor binding domain of SARS-CoV-2 with the receptor could also lead to a breakthrough to tackle this epidemic, which obviously is not that easy, but We pray and hope that they can do it as soon as possible. Fingers crossed. But that's not it. We also need to study more about its other structural components and find out the mutation patterns and the residues which differentiate the SARS coronavirus 2 from the SARS coronavirus 1. By the way, the mutation everyone talks about, like SARS CoV-2 is mutated and all, is mainly in the binding ridge between the receptor ACE2 and the spikes of the virus. It is more complex with additional domain residues than the SARS coronavirus 1. Thank you guys for watching. If you guys have any questions or want to give me any tips on how to make my YouTube video quality better or my channel in general better, just leave a comment in the comment section down below, and I'll see you guys in the next one.